Okay, this video is about chemical reactions in living things and enzymes. So, chemical reactions in living things have to occur in small, precise steps, and they must not give off too much heat, because if you give off too much heat, you may end up cooking a cell from the inside out, which would be bad. Enzymes control chemical reactions and allow them to occur in small, precise steps without giving off too much heat. So, enzymes are proteins, almost all enzymes, but for now we're going to go with all enzymes are proteins. There are some RNA molecules that act like enzymes, but for our purposes we're just going to say enzymes are proteins, and they're necessary for most of the chemical reactions that occur in living cells. A substance that an enzyme works on is called its substrate. And enzymes are specific for only one substrate. So every chemical reaction is going to have its own type of enzyme. Enzymes are referred to as catalysts because they're not changed by the reaction. And so often if you see a chemical reaction, a chemical equation, you'll see the name of the enzyme written above the arrow. And that's also common for inorganic catalysts. Of course, enzymes are organic because they're proteins. Enzymes are used over and over again, not indefinitely, but many, many times before they have to be replaced or are damaged. Enzymes are named after the substance they work on and then end in ase. So, for example, maltase acts on maltose. There are some enzymes that were discovered early on that were, before scientists realized quite how many enzymes there were named before this naming schema came about. So there are some enzymes that don't follow this pattern, but but usually then they also have the name, the more modern name, which is named after the substrate and then the ase ending. Be really careful, because I bet you can guess that the ase ending kind of confuses some students because they see the ose ending, which tells you you've got a carbohydrate. So maltose is a carbohydrate, maltase is the enzyme that acts on it, and enzymes are proteins. And enzymes are produced by cells. Now, remember that if the building block of a protein is an amino acid and an enzyme is a protein, then it's fair to say, and accurate to say, that the building block or monomer of enzymes are amino acids. Enzymes speed up the rate of reactions that would occur too slowly on their own in cells. So here's how they work. On the surface of each enzyme, there's what's called an active site, and the substrate molecule fits this site. Now, the lock and key model would suggest that the fit is perfect. The induced fit model, the enzyme temporarily changes shape and springs back into shape. Kind of like if you imagine you put your hand in a latex glove like the doctors do at the office. It's basically hand-shaped, but everyone's hand is slightly different size and a little bit of different shape. So when the doctor puts their hand into the glove fits the shape of the hand, but then when the doctor takes the glove off, it springs back into its original shape. So that would be like our induced fit model. Now here is an example of an enzyme structure, and this is called catalase. The 4 different colors represent 4 different polypeptides. So catalase definitely has a quaternary level of structure. So when the substrate and enzyme are together, it's referred to as the enzyme-substrate complex. And some enzymes are catabolic, which means they break bonds, and some of them are anabolic. Here is a diagram that shows you a substrate, a larger substrate, and when it is bonded to the active site, it is broken apart, and then the products are 2 smaller molecules. So this would be an example of an enzyme that breaks down a larger product into 2 smaller products. Now, if you get a diagram that you've never seen, every different enzyme and substrate is going to be a different shape. But what you're looking for, if you're trying to figure out which is the enzyme or the substrate, is the enzyme is the one that is the same shape at the beginning and the end, whereas the substrate has changed. So no matter what, whenever you're looking at a diagram that's showing you enzyme activity, the enzyme is the one that doesn't change shape. It's not necessarily the bigger one or the gray one, that just happens to be the bigger one and gray in this diagram. It's the one that doesn't change. So other enzymes may help form bonds, and that would be referred to as anabolic. Now, if you take all of the reactions that occur in our body and you put them together, that is metabolism. So metabolism can be broken into two halves. You've got catabolism, which is the breaking down of things, and anabolism, which is building of things. And again, you can tell that the enzyme is the thing that doesn't change. You've got two smaller substrates that come together and get joined together to make a larger substrate. Now, let's talk about some factors that affect how fast this occurs. So, this next section is all about factors that affect the rate of enzyme action. One of them is temperature. So, each enzyme has a particular optimum or best temperature, and it depends on where that enzyme is in nature. Now, if you look at this graph, you have enzyme A and enzyme B. The optimum temperature of enzyme A is about 35, 37 degrees Celsius, and the optimum temperature of enzyme B would be maybe a little bit more than 60, maybe 61, 62 degrees Celsius. But those are two different enzymes, and it shows they have different optimum temperature. So, what happens above or below that optimum temperature? Well, at extremely high temperatures, the enzymes denature, which means they change shape permanently, no longer function, and once you have denatured an enzyme, you can't get them back. They are warped indefinitely forever. So denaturation has occurred, or you can say the enzyme has denatured. That happens at extremely high temperatures. Sometimes when it gets a little bit past its optimum temperature, if it doesn't go too high and then cools back down, you might have a slight change in the shape of the molecule, but if you don't get it too high, sometimes the molecule will spring back to its original shape. But once it gets denatured, that's it. Now, at extremely cold temperatures, enzymes don't denature. In fact, you can freeze enzymes and thaw them back out, and they will work totally fine. So the reason that enzyme activity is really low at these lower temperatures has nothing to do with the shape of the molecule or that you are permanently damaging them. It has to do with kinetic molecular theory. The idea that if you cool down a system, the molecules will be moving slower and have less energy and collide less often and react less often. It's kind of like being a wallflower at a party and you're sitting in the corner and you're not mingling much and moving around much. You're not going to meet many people. So, it's important for you to note that enzymes and proteins that lose their shape will also lose their function. So, if you cook an egg, that egg white, which is mostly protein, will change shape and be denatured, and you can't get it back. In addition to having an optimal temperature, each enzyme works best at a specific pH. So, I would say enzyme B works best somewhere between 6 and 7, and enzyme A roughly at a pH of 3. Now remember, anything below 7 is an acid, anything above 7 is a base, and 7 is neutral. Now, at extremes of pH, whether it is a very high pH or a low pH, the enzymes will also denature. So here, unlike temperature where enzymes don't denature at a low temperature, but they do at a high temperature, at either extreme of a particular enzyme's optimum, it will denature and not be able to be brought back to its normal shape and never function again. This is why you have to wear safety goggles when you're working with acids or bases in the lab, because your eyes are mostly protein and you don't want the proteins in your eyes to get damaged by an acid or a base. The next factor that affects the rate of the chemical reaction is the amount of enzyme or substrate themselves. So both of these graphs have a similar shape. If you look at the one on the left, you have an increasing enzyme concentration and the rate of enzyme activity. And in this case, the substrate concentration is held constant. So let's say you've got 10 grams of substrate and no more, and you add more and more enzyme. At first, it's going to react faster, but at a certain point, the enzyme is going to run out of substrate to break down, and it won't speed up anymore. And in fact, over time, this curve would actually go all the way back down to 0 once all of the substrate had been acted upon or broken down or built into your new substrate. This is similar, except for the ending would be different. So here, let's say you have a lot of substrate and only 10 grams of enzymes. Now, as you're adding more and more substrate to that 10 grams of enzyme, at first it would speed up the reaction because you've got more molecules to react, but at a certain point it levels off because you run out of enzyme and you have a surplus of substrate and not enough enzyme. This would keep going though until you ran out of substrate, but in here you keep adding more and more substrate and the enzyme is reusable, so this would not go back down to 0. And the last factor I want to talk about is the presence or absence of coenzymes. So some enzymes need a coenzyme in order to function, and those are often vitamins. So if you don't have the coenzymes, the rate of the reaction would obviously be very slow.